Myself Sunita Patra, and in the last 4 sessions we have studied about the structure of protein, what are proteins, their functions, and what are the structural organization such as primary, secondary, tertiary and quaternary structure of proteins. So, and we have also seen the significance, the sources of proteins, from where we can get the vegan diet, the non-veg, from where we can have meat, fresh, milk, we can get. Now, after knowing this sequence, structural organization of the protein, now we have to study how to analyze and to get the linear sequence of amino acids, and how we can get actually this amino acid sequence, what is known as the sequencing of protein. Sequencing of proteins means to get the sequence of amino acid. What is the sequence of amino acid in a particular protein? So, there are certain techniques, there are certain methods which we have to go through it and to study the structure of protein. Now, there are certain so what are the different type of techniques? So, there are Edman degradation, then X-ray crystallography, through which we can study the 3-dimensional structure, the quaternary structure and the tertiary structure of protein. Then Western blotting, the technique Western blotting, which is important for purifying proteins and polypeptides. Suppose when if we are going to study the structure of a quaternary structure of protein, and this protein is having 4 subunits, we have to study all the 4 different isolate all the 4 subunits and to study it individually, for which we have to do Western blotting, SDS page, sodium didecyl polyacrylamide gel electrophoresis, 2D and 2D electrophoresis, isoelectric focusing. So, these are the certain techniques which we go through and which we study the structure of protein. So, determining of amino terminal sequence detection. Now, prior to sequencing peptides, it is necessary to eliminate the disulfide bonds. Now, we know that suppose if the protein is of tertiary structure. So, this tertiary structure of the protein contains some of the weaker bonds and also the disulfide linkages. So, weaker bonds can be disrupted, but if the peptide it contains a disulfide linkage, then it should be eliminated with the peptides, between the peptides, within the peptides, and suppose a polypeptide chain can contain disulfide linkage within itself and also between the peptides. If there are a quaternary structure of protein and both of these polypeptide chains they are sharing the disulfide linkages, because for determination of for sequencing of amino acid we require a linear chain of amino acids. So, all the bonds which folds the proteins should be disrupted. Now, several different chemical reaction can be used in order to permit separation of polypeptide strands and prevent protein conformation that are dependent upon disulfide linkages. So, there are chemical reaction which are used to permit the separation of peptide strands. Suppose if they are of quaternary structures, so we have to separate the strands, both of the strands, or we have, if we are doing secondary structure beta pleated sheets. So, we have to separate the strands and then prevent protein conformation that are dependent upon disulfide bonds. Now, how we can do this, how it is done? So, this is accomplished by cleaving the protein with a proteolytic enzyme, say such as a trypsin, so or a chemical reagent such as cyanogen bromide. Now, these chemicals they generate a set of peptides. Suppose if it is a long polypeptide chain, so what we have to do? We have to cut this polypeptide chain into peptides, a set of peptides, or we can say fragments of the original protein. So, we can actually sequence the peptide, the sequence of amino acid in different fragments that can be separated and then be sequenced. So, in spite of taking overall the polypeptide at a time, these polypeptides they are cleaved either by trypsin or by cyanogen bromide to get a set of polypeptides or fragments of the original proteins and that they are separated and then they are sequenced. The purified sample of peptide, what we have to do first, it is hydrolyzed by a strong acid at 110 degrees centigrade for say about 24 hours, and then what happens when we do this experiment, we digest this protein or hydrolyzed by a strong acid for more than 2 hours of 24 hours, it cleaves the peptide bond and releases the individual amino acids. Now, these amino acids are separated by chromatography. So, first of all what we have to do, first we have to purify the polypeptide and then we can go for further cleavage process. Now, there are three major chemical techniques for sequencing peptides and proteins from N terminus, because we know that the primary structure of protein, it has a N terminate N and C terminate N, and we are going to sequence protein through N terminus N. So, there are three major chemical techniques. These techniques are Sanger's techniques, Dancyl chloride and Edman techniques. Edman degradation, we are talking about Edman degradation. Now, why we are taking Edman degradation? The utility of Edman degradation technique is that it allows for amino acid sequence to be obtained from N terminus inwards. Suppose we are going to sequence amino acid, that means we want to get a sequence of amino acids. So, we will start with N terminal end, and Edman degradation methods, it helps in sequencing protein in N terminus region. So, using this technique, this method, it is possible to obtain the at-dry sequence of proteins. So, gradually one by one amino acid which is attached with that N terminal end, it can be obtained, it can be categorized, it can be seen, and then gradually one by one we can obtain the entire sequence of polypeptide. This method utilizes phenyl thiocyanate to react with the N terminal residue under alkaline condition. So, this is the first step. In the first step, what actually it is done, the polypeptide and phenyl thiocyanate it is used, and in the phenyl thiocyanate it interacts with N terminal residue of amino acid, and the condition is alkaline, in alkaline condition. What actually happens in alkaline condition? So, the reaction in alkaline condition, it results in the rearrangement of the released of N terminal residue to a phenyl thiodantoin derivatives, and then added advantage of this Edman process is that the remainder of the peptide is intact. That means, suppose if the polypeptide chain is having say about 15 to 20 residue, and the first amino acid is going to react, and then it reacts, and then there is a process of rearrangement and then release. So, N terminal residue to a phenyl thiodantoin derivatives, then rest of the derivatives, rest of the residues of the polypeptide chain, they remain intact, they remain in the same in the polypeptide. The rest of the polypeptide, it is intact. The entire sequence of reaction can be repeated. So, first what we have to do, we have to do the same sequence of reaction repeatedly for each and every coming amino acid. So, one by one we have to repeat the same reaction over and over, so that we can obtain the amino acid sequence for the whole peptide. So, this process can also be now this process has been automated. Initially it was not automated, but now we can automate this sequence, and this automation, it allows the rapid and efficient sequencing of even extremely small quantities of peptides. So, automation it is very effective and efficient in the process of sequencing. So, here we can see in the region that how the first phenyl isothianate, it reacts with the first residue, the terminal N terminal residue, and then there are three steps: first reaction, then release of first amino acid, and then again a repetition. So, again and again all the three steps, they are repeated one by one. So, that till the end of the peptide, how many amino acids they are attached, or they are present, or they constitute the polypeptide chain. So, they are analyzed and characterized one by one by the repeated these three steps, the repetition of all these three steps. So, again here it is shown uh, the coupling, and then cleavage, and then conversion. These are the 3 steps in Edmonds reaction. So, first the coupling of the reaction, then there is cleavage of the first amino acid, and then to give a stable structure, and then conversion, and this converted structure can be separated, and after separation it can be analyzed what type of amino acid. So, all these 3 steps, they are repeated again and again to get the whole sequence of amino acid. So, the basis of the method peptide sequencing by Edmond chemistry may be divided, as I have already said, that it can be divided into steps. All the 3 sequences, it can be divided into steps. Now, what are these type of steps? So, the strategy of Sanger sequencing, and then it is also known as Edmond degradation method. The strategy of Sanger's for the sequencing of insulin was to characterize the series of small overlapping peptides. So, overlapping, if we are going with overlapping peptides, we can arrange. If we are getting the peptide, it is very difficult to arrange the sequence of again the amino acid or the peptide. So, the arrangement should be overlapping, so that we can sequence, we can get the whole sequence of amino acid in a particular polypeptide. So, overlapping to produce a cleavage of the perfect molecule of the parent molecule. What are the parent molecules? So, we get our overlapping peptides which are produced by cleavage of parent molecule, and determination of the overall amino acid content, and identification of the N-terminal residue for each peptide is allowed, and detection of the sequence of the whole molecule, it is done sequentially. Now, there is an alternative approach. The Edmond has also given the same approach, and in 1950 this allowed the determination of extended sequence of peptides or the whole protein. Now, this method employs chemical reaction to remove and identify the amino acid residue, that is the N-terminal of the polypeptide chain, that is the residue from a free amino acid group. Now, this step is repeated, as I have already explained the process. This process is repeated so that we can get the sequence of polypeptide. So, we are sequencing one by one amino acids from the amino terminal residue. So, this is the main basics of this method. Now, this is the key. What is the chemical degradation? What we are doing? What is the chemical process? It is the phenyl isothianate which reacts with amino acid. We have already seen in the previous slide with the amino group, or in the case of polyl residue with an amino group at the N-terminal end of the polypeptide chain to form a phenyl carbonyl derivative of the terminal residue. So, we are reacting, we are giving the polypeptide chain, the phenyl thiocyanate will ultimately gives phenyl carbonyl derivative of the terminal residue, terminal amino acid. So, this reaction is known as the coupling reaction, the coupling of phenyl isothianate with that of terminal derivative of the amino acid. The next step, now the basic condition required for this reaction are Edmond originally used pyridine to generate a pH of 8.6. Clearly, a free amino group is required for this reaction to occur. If the amino group has become modified and will be no longer react with PITC, the polypeptide is said to be blocked. So, we have to get a free amino group. So, main requirement for this is a pH of 8.6 and clearly a free amino group. If the amino group is blocked or it is modified, it is not going to done. So, PITC will not react with this, and the polypeptide, the further sequencing process is disrupted. The polypeptide chain is blocked. Now, in the presence of strong acid, first was the coupling reaction, then second step is the cleavage. So, in presence of strong acid, the cleavage occurs at the first peptide bond, because we have to sequence amino acid one by one. So, this strong acid, it cleaves the first peptide bond, giving the peptide minus the first residue. The rest of the residue is of the peptide, it is intact. So, minus the first, and liberated the first one residue as the ATZ in ATZ form, anilothiazolinone, it is ATZ, abbreviated as ATZ form. So, first was coupling, second was cleavage and removal of amino acid, the first residue in ATZ form. Now, once the reactants and the products have been washed, again now what we have to do? We have to we have to do the coupling reaction, then cleavage reaction, and then when the products come out, we have to wash the reactants and the products. The products are washed away and the shortened polypeptide chain. Now, the polypeptide chain -1, so because the terminal one has been cleaved out. So, the shortened polypeptide chain can be taken through the another round of coupling and cleavage. So, coupling, cleavage and modification. So, coupling, cleavage, it modifies, it continues after the washing, the wash away. So, coupling and cleavage to release the second residue, and so on in a cyclic fashion. The coupling, cleavage and removal, washing, it goes on till we reach the end of the sequence, till we get the all the sequence of amino acids. Now, third is conversion. So, coupling, then cleavage, and third one is conversion. The ATZ residue which is separated, which is washed. Now, what we have to do? We have to cleave all the residues, all the amino acid in the ATZ forms, and now after getting this amino acid, what we have to do? This ATZ residue is separated from the polypeptide by extraction in an organic solvent, ethyl acetate or chlorobutane. So, these are the solvents, organic solvents which we can get, and is then converted to a more stable form to allow better analysis. Now, we have to analyze what type of amino acid it is. So, we have to convert this form into a more stable form, which can be done by treating it with ethyl acetate or chlorobutane. The conversion to more stable form, phenylthiohydratone PTH form, is done in the aqueous acid 25% FTA volume by volume in water. So, conversion, so first is coupling, second is cleavage, third one is conversion, and after conversion we have to move it into a more stable form, which is treating with PTH. Now, analysis of PTH residues. Ultimately, what we have to do? We have to characterize each and every amino acid, and PTH residue is most stable one. So, coupling, cleavage, conversion, and then convert into a more stabilized form. The PTH residue generated by each cycle of Edman chemistry is typically identified by chromatography. Now, if we have to isolate, identify the amino acid, and originally by thin layer chromatography, lately by reversed phase high performance liquid chromatography. These are the technique by which this amino acid, which has been cleaved and is converted into more stable form, we can use this chromatographic technique for further analysis of each and every amino acid of this polypeptide chain. Now, structure determination. So, this was the sequencing of amino acid. Now, how can we determine the structure? What are the strategies which we can determine the structure? So, various functions of biological systems, they totally depend upon the structure of protein, because structure always is according to the function of the protein. If the structure of protein is deformed, the function is also affected. So, determine the structure and function of proteins, it is actually the task of the protein. We have to determine the structure of protein and accordingly. So, characterization of structure and function of protein, we have to do undergo certain process. We have to see the 3-dimensional structure and dynamics of proteins, and to understand the function of protein at molecular level. So, it is necessary, molecular structure, it is necessary to determine the 3-dimensional structure. If we want to analyze the function of protein, we have to analyze the protein in its 3-dimensional residue, its 3-dimensional structure. So, determination of secondary, tertiary and quaternary structure of proteins can be done by X-ray crystallography or by nuclear magnetic resonance spectra of proteins. So, we have to use these techniques to determine the structure of protein. What is X-ray? The tertiary structure of protein can be detected by X-ray diffraction, spectrophotometry, electrophoresis and chromatography. So, these are the techniques by which we can get this tertiary structure of protein can be detected. Now, about X-ray, what is X-ray crystallography? So, X-ray crystallography in turn is a very high molecular microscopy. So, what actually what is the principle behind X-ray crystallography? It is based on the fact that X-ray are diffracted by the crystals. So, the way the atoms they get refracted by the crystal, they can form a 3-dimensional structure of the protein. So, to find a 3-dimensional structure of protein, we have to undergo the X-ray crystallography to enhance or to how the protein function, what are the functional domains, what are the 3-dimensional structure. It enables to visualize the structure. So, X-ray crystallography is based on the diffraction of the X-rays by the crystal, protein crystal. So, molecular size, because of size of molecule is too small to see under a normal light microscope. So, or against a microscope, what we have to do? We have to the molecule must be much larger in molecular size. So, what we have to do? We have to go through X-ray radiation to look into a molecular level, its wavelength is much. So, X-ray crystallography, what is the principle? As we have already seen that the X-rays are diffracted by the crystals and direct detection of atom position in crystal can be done. So, this is the principle. In the diagram we can see that the X-ray it is get diffracted by the crystals, and by this diffraction the direct detection of atom position in crystal can be determined. Now, there are certain technique which is known as Western blotting of technique. Western blotting means what is the system of blotting, because this blotting has been applied in both DNA, RNA and protein. So, to immobilize a protein, suppose we have first we have done a process of electrophoresis and we have run the protein in SDS page, and there is separation of protein in the form of a batch, and now we want to transfer that protein from gel to blotting papers in the nitrocellulose paper. This technique is known as blotting technique. So, when it is done with DNA, it is known as southern blotting, and we also use RNA for detection, for separation from different molecular segments, peptides. When it is a protein, it is known as Western blotting. When it is RNA, it is known as northern blotting. So, northern blotting, southern blotting, northern blotting and western blotting. The technique is the same, but when we use when this system, this technique is used for protein, it is known as western blotting. Now, this is the technique of western blotting. What we have done? We have we got a sequence after gel electrophoresis. After gel electrophoresis, the stack of nitrocellulose paper is placed there and the gel is also placed there, and now this gel, this bands, protein bands which we can see in the gel is they are transferred to the nitrocellulose paper. And when we get this sequence, now what we can do? We can raise antibody, we can raise a marker, we can raise a probe, body probes to detect a particular protein or a polypeptide chain through autoradiography. So, after electrophoresis, the bands, the protein, they are transferred to the nitrocellulose paper. So, this technique is known as western blotting, which is further used for the detection of specific polypeptide chain with the help of autoradiography. So, molecule of interest is a protein, and now how we can detect? So, the molecular of interest is a protein, we are dealing with the protein, and what is the probe? The probe is the antibodies. So, because the molecule is a protein, so we can raise an antibody against this protein, that particular protein. So, probe, what is probe? It is an antibody which has been raised against that particular protein which we want to detect. Now, what are the prerequisite? The SDS page is prerequisite for western blotting. So, what we have to do, as I have already said that the protein bands, they have to transform from the gel to the nitrocellulose paper. This process is known as blotting. So, for this we need for protein in generally the DNA we use agarose gel electrophoresis, and proteins we do sodium deutesyl polyacrylamide gel electrophoresis. So, first we have to perform the the SDS page so that we can get separate the proteins of different molecular weight, and after that we can go for Western blotting. Now, this is the transfer. The first what we have to do, we have to run the proteins in SDS page, we have to perform that function and then transfer the protein. The proteins moved from gel to the membrane which is made of nitrocellulose, and now actually we can also go for electroblotting. Electroblotting because it requires much lesser time. So, within 2 or 2.5 hours the bands can be transferred from gel to electrophoresis paper. And then blocking. Blocking is placed by membrane in a dilute solution of protein with a minute amount of detergent. We have to block, we have to immobilize the block is achieved. Now, this is achieved by placing in dilute solution of protein with a minute amount of detergent. So, first what we have to do, first we have to go for SDS page, then Western blotting, that is transfer of protein from gel to nitrocellulose paper, and then the blocking process. Now, then after blocking process, then what we have to do, we have to detect. How can we detect? We can detect it by a probe. And what is a probe? It is an antibody which has been raised against the protein of question, the protein which we want to analyze. So, detection, a modified antibodies is used which is linked to a reporter enzyme, and when exposed to the substrate it produces a color. That means, we have to use a chromogenic substance which we can morphologically, which we can analyze by changing. Suppose if a probe is an antibody, it reacts with the protein of interest, detect a molecule. So, there is a chromogenic changes, there is a color formation, the product is produces a color, so that we can identify the protein. So, it can be detected, and then analysis. After detection it is analysis. And now, what is the advantage of all this? It is specific test for detection. Suppose for an example we can use this test for HIV to compare from other tests. It tells us how much protein can be accumulated inside the cell. If we want, we can also analyze that how much protein can be accumulated inside the cell. It gives the information about the size of proteins. It can detect now one protein mixture. Suppose there are protein mixture, suppose we have 4 to 5 protein in a mixture. First what we can do, we can go all these proteins, they have different molecular weight. So, first we have to run SDS page. So, then all the proteins they can be separated, they are separated by their different molecular weight, and when they are separated we have to go for Western blotting, and then Western blotting, then we can go for detection for analysis and autoradiography, and then further we can go for its molecular size determination and all that. And what are the application? It can identify the nature of the protein. It can be applied as a tool of quantitative analysis of the micromolecules antigens, cooperation with immunoprecipitation. So, there are n number of applications what we can do with this. The Western blot is extremely useful in biochemistry for the qualitative detection of a single protein. Protein modifications such as post translation modification can also be determined. We can see that proteins when they are formed a linear sequence of an amino acid, and when if the conformation is changes, suppose it changes into a 3-dimensional structure, a tertiary structure. Now, after saying after this conformational changes, there are certain groups according to the function the protein it is modified. This modification is known as post translation modification. Post translation means after synthesis of protein, these proteins are modified. Some of the glycohydrates, suppose we were saying that the conjugated proteins. So, conjugated proteins which contain certain proteins, certain carbohydrate moieties, certain phosphate group, sulfur group, sulfate group, whatever which is required for its function, it is modified. So, these are post translational modification. After completion of protein synthesis, they are modified. So, this type of post translational modification can also be determined by the Western blotting technique. Molecular diagnostic application, the viruses is enveloped with different proteins. So, detection and presence of this protein is useful for the detection and presence of viruses. Suppose because the virus contains of capsid protein and the DNA part. So, by detecting the capsid protein part, the protein part of that virus, we can easily identify the presence of these viruses. So, these forms they are used for disease diagnosis. Now, we were talking about SDS page. What is SDS page? So, SDS is sodium deuteryl transferrin. It is also called lauryl sulphate. It is an anionic detergent. Why it is used? Why it is used in polyacrylamide gel electrophoresis? Because when this protein it is dissolved with SDS, that it dissolves its molecule and it has a net negative charge within a wide pH range. So, what we have to do for electrophoresis? What we have to do? We have to make the protein molecule or all negatively charged. A polypeptide chain, what actually happens? A polypeptide chain binds the amount of SDS in proportion to its relative molecular mass. So, what happens? The negatively charged SDS destroys most of the complex structures of the protein and are strongly attracted towards anode. So, the protein which is to be identified, it gets a overall net negative ion. So, it moves towards anode and the all the bonds which are formed, it comes in a linear fashion. So, the polypeptide chain first of all it binds to SDS and then it has overall a negative charge, and so that all the proteins now they run towards anode positively charged, and so that and then according to their molecular weight they get separated in polyacrylamide gel electrophoresis. So, polyacrylamide gel electrophoresis it is of two type 2D gel electrophoresis, single dimensional electrophoresis. Then we can rotate the slab into 90 degree and we can go for second 2D or it is 2 dimensional electrophoresis. It is a key technique for purifying individual proteins from complex samples. If we have a complex sample of protein and we want to identify all the proteins, we have to do 2 dimensional electrophoresis, which is based on the isoelectric point. So, it is very important. The first electrophoresis SDS page it was done, and the purification of the protein it was done according to the charge and the molecular weight. Say about if the two proteins or the two polypeptides they are of same molecular weight, they cannot be separated by single dimensional electrophoresis. Then what we have to do? We have to do 2D gel electrophoresis, and now in this the proteins or the polypeptides they are they are separated depending upon or based upon the isoelectric points and the molecular weights. So, each and every proteins they are separated according to their molecular weight. There is a pH gradients and the isoelectric point, isoelectric focusing when the protein when it runs through this gel, there is a neutralizing point where the protein it becomes stagnant and it separates. So, two key points are molecular weight and isoelectric focusing in the second dimension 2D electrophoresis, 2 dimensional electrophoresis. So, what is isoelectric focusing? It is a type of zone electrophoresis. Electrophoresis is separation of charged molecules or the particles, which we have already done, and ions in the liquid medium under the influence of electric field. So, electrophoresis is separation of molecule under the influence of electric field. It deals with separation of amphoteric compounds. Separation is done or is achieved by applying a potential difference across the gel that contains a pH gradient. So, isoelectric focusing is what it is separation. In this process separation is achieved by applying a potential difference across the gel that contains a pH gradient. So, the protein molecules we can see here the protein molecules, this is the technique how it is done. So, pH gradient is maintained throughout the gel, and the proteins or the polypeptides they are separated by isoelectric point and by the pH gradient. So, these are the techniques which we used in detection of protein. So, when we get a protein, we know we have to, if we want to know the function of that protein, we have also to study the structure of the protein. So, structure of protein can be done first 3-dimensional structure of protein. If you want to know the function of protein, we have to go through the X-ray crystallography. So, X-ray crystallography, it performs, it enables us to analyze the structure of protein, and then after analyzing the structure of protein or the quaternary structure of protein, if we want to analyze the individual polypeptide. So, what we have to do? We have to separate all the polypeptide. If the protein of the polypeptide is of a longer length, what we have to do? We have to cleave, what we have seen in the trypsin digestion. So, we have to go for trypsin digestion. After trypsin digestion, what we have to do? We have to do for SGS page electrophoresis. In this, the protein molecules, they are separated on the basis of molecular weight and ion, and after that we have to go for 2D electrophoresis. 2D electrophoresis, that in which the proteins, they get separated by the isoelectric point and the molecular weight. Then we can go for Western blotting. Western blotting, the proteins, we can take transfer the proteins from gel to nitrocellulose paper, and then in nitrocellulose paper, with the help of the probe, the antibodies which has been raised again. If we want to particularly analyze a particular protein, we have to raise antibodies against this, and this is bind to a chromogenic substance. So, after binding in after this Western blotting, the probe, the antibodies is bound to it, which gives a color which recognizes that protein, and thus we can analyze the structure of protein, the amino acid, and then what are the polypeptides, and after getting the polypeptides, we can go for the sequencing. So, that we can get each and every knowledge in right from the structure, the function till the amino acid sequence of a particular protein. So, it can be detected. In this way, we can get the complete knowledge about the protein. So, thank you. So, I have taken again the acknowledgement are due to unknown and known sources which has been used for making this presentation, and without which this presentation or this information or this data information, the presentation would not have been so informative. Thank you once again.